into our question and answer period. And thanks to all who've already submitted questions. I have a kind of a basic and general one to start. What in short, what is the difference between hazard and risk assessment at RIFM?
Thank you for that question. It's a really good question, Laurie. So hazard identification, in essence, addresses whether the chemical is a sensitizer or not at all. That's the hazard identification. Risk is hazard combined with exposure. Is it safe at what level? That's sort of the risk assessment that we do.
Thank you for that. Nikita, we have some questions for you. One is, are there any validated in vitro tests available for respiratory sensitization that are accepted by regulatory bodies?
At the moment, for respiratory sensitization, we don't have anything that's validated or accepted by the regulatory bodies. There are models under development. You'll find that complex tetra or co-culture models are available, but they are all under development. And so it's a field that is new. We are working on it. And yeah, I mean, it's unfortunately at the moment, nothing is either validated or accepted by regulators.
Thank you for that. We'll have to stay tuned, right? There was a very detailed question having to do with the cleaning industry. And it goes something like this. Do you foresee any model that can be leveraged for potential respiratory sensitizer, in particular enzymes and microbes used by cleaning industries as a spray? And I'll let you address that one.
Well, I think for high molecular weight, when you're talking about enzymes, the high molecular weight respiratory sensitizers, there's already some understanding available that you can evaluate them in a way. But the focus that our research at RIFM here is on low molecular weight respiratory sensitizers, where a lot of the information is still very limited, mainly because of the limited chemical space. Like recent publication we had in critical reviews in toxicology, we highlighted that based on the available occupational data from human exposure to the respiratory sensitizers, we could identify less than 10 with compelling evidence of respiratory sensitization that had strong supporting medical evidence from that were evaluated by professionals in that field, medical professionals in that field. And so that's a very low number of respiratory sensitizers to work with. And yeah, it's hard to say if there is any particular model that would be best suited for the purpose that you're asking. But I would assume that if you look up for the high molecular weight respiratory sensitizers literature, you'd find something in there. I mean, I can't speak to that yet. It's, as I said, like our focus is more on low molecular weight respiratory sensitizers.
Thank you. We have a lot of questions pouring in. Thanks, everyone. Here's one for Miwa. What can you tell us, what processes are being taken to assess or evaluate the sensitization potential or potency of new extracts isolated from plants that might have limited chemical composition or history of use information? So Miwa, what can you tell us?
Thank you for that question. That's still an area where much research is still needed. We are at the moment depending on the available composition for natural complex substances safety assessments. But I think the new in vitro assay such as Senses, in which the whole substance can be literally put on the reconstructed human skin, those can be promising assays to look at. But at the moment, we would need the composition data to assess the safety.
Oh, good. And one person says, thank you for this great presentation for both of you. And is there a similar effort at RIFM on ecological impacts of fragrance ingredients, such as aquatic toxicity? If so, a presentation on that would be very interesting. So is there anything about, is there a similar effort at RIFM on ecological impacts? I wonder if you or Nikita wants to answer that one.
Go ahead, Mia. That's fine.
We do have an environmental toxicologist, Aurelia, at RIFM, and she would be able to give you details on ecotoxicology.
Oh, good. Okay, thank you. Here's another question for either of you. It's about read across. What are the parameters? What are the parameters in structural analog, as in percent similarity, uncertainty factor, TK and TD data consideration? And can we use 2 or more fragrances in a single finished product from a skin sensitization perspective? So that would be for you, Miwa, to try to tackle.
Sure. For the read across, we do look at structural similarity a lot. We do look at Tanimoto score as well, but we do take into account with heavyweight the expert judgment from our expert panel chemists. So looking into the metabolism breakdowns and judging based on their chemical structures, that will be our strategy for read across. We did outline the detailed process of choosing read acrosses recently in our publication. Maybe I can put that in the chat soon as well.
Okay, great, thanks. And our speakers have said they can go a little bit longer today than the top of the hour, so we'll keep going through the questions. Thanks again, everyone. Here's a question for either of you. How do you differentiate the assessment when the same substance is used as a fragrance and as a flavor? Do you get into that? How do you differentiate?
I could start. So yes, there is an overlap between the flavor and fragrance ingredients in this industry, but I think the key distinction is the source of the exposure values that we get. The Creme RIFM model that takes into consideration the type of products, I think they kind of distinguish between the use of that ingredient as a flavor versus as a fragrance ingredient. But in the end, I think depending on the endpoint, if there is data available that we can use to make the evaluation, we'll use it. So if it's oral exposure, that goes into the systemic toxicity evaluation. If it's airborne, it's inhaled, it goes for respiratory exposure evaluation. If it anything that goes into the environment, then that gets under the environmental endpoint. Miwa, would you like to add anything to that?
Yeah, I think regardless of whether it's being used as flavor ingredient or not, if it has odor qualities, we do assess it and consider it as a fragrance ingredient.
Oh, good. Here's another skin question for you. Someone would like to know, how do we arrive at 1 milligram CM, do you say squared, test sample application in a skin sensitization study? Is that someone who should contact you, or is there an answer for that?
Yeah, I'm not sure if I understood the question correctly. How do you achieve 1 micrograms per centimeter squared of the material on skin?
I believe so. How do we arrive at that?
Okay, so for that, you would need to know the percentage of the working solution. So what percentage of this fragrance material is in your vehicle, plus the size of the patch. So given those, you can do the area calculation.
Ah, thank you. Here's another question for you. Has RIFM evaluated or used the skin sensitization potency assessment in the OECD guideline 497, specifically using some specifics and in silico? Anyway.
Yeah. So we can do that. Definitely, we are able to do that. But what we see is that the fragrance ingredient inventory, if they are sensitizers, they're either weak ones or very weak ones. The methods outlined in the OECD 497, they're really good at identifying strong sensitizers, which we don't see often in fragrance inventory. So that's why we're exploring into other methods that can give us the potency determination in the weak and very weak area.
Good. And someone wondered, is the skin sensitization concentration a dependent phenomenon?
It's a threshold phenomenon.
It's a
Yes.
threshold.
Dose dependent, yes.
Thank you. And we have one for Nikita. Thank you. Also, are the quantitative risk assessments, the quantitative ones, done manually or with the use of any software or AI-based models?
The whole process is manual.
The process is manual today.
Yes. So as I mentioned, the 4 steps, we do literature search using different databases. We have our own database at RIFM. We also tap into the ECA database and EPA's HPV and HSDB as well. So anything that's out there available that can help us make an evaluation in terms of a study that's already done, or in the near future, we would also be utilizing in vitro models towards this end as well. So it's a manual process as of now. It involves a lot of expert judgment. You have to look at the effects, evaluate them, make informed decisions about what is the effect level that you're seeing. Is that the true effect level? Do you think the species sensitivity is factored in there as well? So transferring that data from animals to humans, that is not always a straightforward process. So yes, it involves a lot of manual input and expert judgment.
Oh, okay.
And I just wanted to add quickly. So this is where we might be able to help you guys. We have already done this work and we have automated a lot of data provisioning for the different endpoints and for the different types of chemicals. We should talk further.
Yeah, great. And that was from Neelam Vaidya, Viridis-Chem CEO. And thanks to our speakers for talking a little bit extra today beyond the top of the hour. Here's another question. How do you assess the hazards for fragrant materials that are extracts or mixtures? For example, in the case of a botanical fragrant extract, when there's no human or animal data, and there could be hundreds of phytochemicals in it, would an SAR approach be valid for that? Who wants to answer that one?
I could take a stab at it. So as per the process that we have just laid out, we are just getting started with evaluating safety for NCS. I think the go-to step for us to ensure that we are looking at a list of components within that complex mixture. And so far, we have done fragrance ingredient safety assessment. So obviously, our first step is to consider that all those fragrance ingredients or the fragrant components of that mixture are okay. And then I think the next step from there would be to look at those other components that are not fragrances. So it's a developing process for us at the moment. We have laid out a base that we are starting on, and that involves looking at the components. So it's not an unknown. It will be known, I think, up to 0.1% of the component concentration in that mixture. So I guess a lot more details are available on the publication that just came out earlier this year, on the natural complex substance safety evaluation criteria from RIFM. Okay. So just to give you a gist, we do look at the components down to 0.1%, so they're not totally unknown, and we are devising the plan to look at all of those ingredients or all of those components. Okay. Niva, would you like to add anything?
No, I think you captured it.
Okay, and we have many more questions, so we'll keep going for a little bit. How would a QRA be conducted in the future based on systemic toxicity? Anyone want to tackle that one?
I can take that one. So it is sort of already being accounted for in our QRA2 process. So what we would do is to set the maximum acceptable concentration based on skin limits, skin nestles, and compare that to what's allowed by the systemic NOELs, the reference doses. And we would take the lower one to officially set the limit for that material.
Oh, good. And someone wondered she wanted to hear about the synergistic effects between fragrance and other ingredients, such as surfactants or preservatives. Any comments there? Is
I think
relevant?
Yep. So I think that would be very dependent on the endpoint that you're looking at, whether it's a hazard-based endpoint or a threshold risk-based endpoint. For skin sensitization, which is an immune response, we have never come across a case where a synergistic effect became an issue. So we do our risk assessment under the assumption that each component can be assessed on its own.
Okay.
And I would like to just add to that, when we are looking at NCS, that's exactly what Neva said, but looking at fragrance ingredients versus other ingredients in a complete or a final product formulation is something that we don't do. We only focus our assessments for fragrance ingredients and NCSs. So a full formulation would be at the end of the company whose product it is.
Okay, thank you. And here's another skin question. Does RIFM have any perspective on the elicitation threshold for a known skin sensitizer?
So not so much, because our goal is to prevent people from obtaining or getting sensitized. So once you're sensitized, our suggestion is to consult with the dermatologist and avoid that material.
Got it. Okay. Anything really crucial that we haven't asked about that either of you wants to talk about briefly, and then we'll wrap it up? Or do you feel like we covered the main stuff?
Yeah, no, this has been great. Thank you. This was a good opportunity, a great discussion. And for any of the attendees, if you still need need to discuss more in detail or have any more burning questions, please do feel free to reach out at RIFM, either of us or any other endpoint scientist. The details you can find on our website. So thank you.
Great.
Yeah, thank you. Thank you for this opportunity to talk about our risk assessment process. And
okay.
Yeah, same. Please reach out if you have any questions or comments.
Great. Well, thank you. The thank yous and compliments are flowing in. And on behalf of VeritasChem, we want to thank you both. And thank you to everyone who attended today and to everyone who watches this recording. And for more information about VeritasChem, you can go to veritaschem.com.
I would like to just add that thank you, Laurie and Riddhi, for making this webinar a success. You have always been great. Thank you.
Thank you so much, Neelam. Thanks again, everyone. We'll say bye for now and hope to see you at future VeritasChem webinars.